Hello students, welcome to EPG Pathshala. I am Dr. Sudhita Gautam Parihar from Hansraj College, University of Delhi. Today we are going to discuss about a module, Principles of Gene Expression, Transcription under paper Molecular Genetics. After studying this module, you will come to learn about the following module explains the central dogma of molecular biology, the basic differences between DNA and RNA, purpose of transcription process in biological system, and the three events initiation, elongation and termination. How the genetic information which is coded in the DNA is converted to RNA before translation can begin. Differences between prokaryotic and eukaryotic transcription during the three events, that is initiation, elongation and termination. The word transcript means written or printed version of something. Transcription is a vital process of the biological forms having a complex regulatory system. In 1953, Watson and Crick gave the double helix model of DNA, and 3 years later, Crick gave the central dogma of molecular biology, which stated that genetic flow of information within different organisms is a stepwise process. The genetic information stored in DNA is activated to give mRNA through transcription, and from mRNA finally to proteins through translation. Where does this take place? In the cell. What is central dogma? What is the significance of central dogma? It was first given by Francis Crick in 1958. DNA was first extracted by Frederick Mischer in 1869, and its molecular structure was first identified by James Watson and Francis and Crick in 1953. Central dogma of molecular biology. The central dogma of molecular biology is the process of flow of genetic information within a biological system. It was first given by Francis Crick in 1958. In other words, central dogma, protein information cannot flow back to nucleic acids. Significance of central dogma. The central dogma of molecular biology explains that DNA codes for RNA, which codes for proteins. In the central dogma, you can learn about the important roles of messenger RNA, transfer RNA, and ribosomal RNA in the protein building process. Difference between RNA and DNA. Characteristics: DNA, RNA, sugar, deoxyribose sugar in DNA, ribose sugar in RNA, nucleotides, adenine, thymine, guanine, cytosine in DNA, adenine, guanine, uracil and cytosine in RNA, strands, double-stranded in DNA, single-stranded in RNA, presence of 2' hydroxyl group present in DNA, absent in RNA. Stability: more stable DNA, less stable RNA. Types: A, B and Z, DNA, rRNA, mRNA, tRNA, snRNA, miRNA, siRNA for RNA. Structure of ribose and deoxyribose sugar. Types of RNA. Depending upon the type of function, the RNA molecules are classified as type of RNA, function, location. First, ribosomal RNA, RNA. Function, structural component of the ribosomes. Location, cytoplasm. Type 2, messenger RNA, mRNA. Function, carries the information in a gene for the protein synthesis. Location, nucleus and cytoplasm. Third, transfer RNA, tRNA. Function, transport amino acids to the ribosomes during protein synthesis. Location, cytoplasm. Fourth, small nuclear RNA, snRNA. Function, modification of the RNA transcript. Location, nucleus. Fifth, microRNA, miRNA. Function, RNA interference. Location, nucleus. Sixth, small interfering RNA, siRNA. Function, RNA interference. Location, nucleus. The transcription process. The genetic code of DNA genotype is passed to RNA through the process of transcription. It requires a series of events involving various RNA nucleotides, DNA template, and a series of protein components for its initiation and regulation. Transcription process requires three major components. First, RNA polymerase and associated protein factors. Second, DNA template. Third, raw materials or the nucleotides. RNA polymerase, catalyzing the process of transcription, was discovered independently in 1960 by Samuel Weiss and Gerard Hughes. The information on the DNA is used by RNA polymerase to make mRNA using one of the strands of DNA as a template, also known as the antisense or non-coding strand. The double-stranded DNA is unwound. One strand to which the RNA polymerase attaches acts as the template strand. The other strand is referred to as the non-template strand, coding strand, or sense strand. Transcription begins on specific DNA sequences called promoters. It occurs in three phases: initiation, elongation, and termination. Transcription, process of copying DNA to RNA, differs from DNA synthesis, in which only one strand of DNA, the template strand, is used to make mRNA, does not need a primer to start, can involve multiple RNA polymerases, is divided into three stages of initiation, elongation, and termination. DNA acts as the template for transcription. In 1970, Oscar Miller, Barbara Hamcallo, and Charles Thomas provided the evidence of DNA molecule being used as a template for transcription. Electron microscopic studies of internal cellular contents revealed presence of Christmas tree-like structures, thin central fibers, the trunk of the tree, to which were attached strings, the branches with granules. When deoxyribonucleases were added to it, breakdown of the central fibers was observed, indicating that the tree trunk were DNA molecules. Ribonucleases removed the granular strings, indicating that the branches were RNA. The Christmas tree-like structures was concluded to be a gene undergoing transcription. As the process of transcription proceeds, more and more RNA is formed, which further extends the branches of the tree. DNA molecule unwinds to act as a template, that is, the double helix opens up and only one strand acts as a template for the transcription. This is the strand to which the RNA polymerase binds and transcription process continues, which is exactly opposite to the coding strand, which contains the gene sequences. As transcription takes place on this template, the new strand is perfect replica of the coding strand of DNA molecule. At a given time, only one of the DNA acts as a template. One strand of DNA acts as a template for transcription. In 1963, Julius Marmer and his colleagues proved that only one strand of DNA acts as a template. They made use of DNA bacteriophage sp8, which infects the bacterium Bacillus subtilis. The double-stranded DNA of this phage has different densities for each strand, which permits the separation of the two strands by equilibrium density gradient configuration into heavy and light DNA strands. Bacillus subtilis was placed in a medium containing a radioactively labeled precursor of RNA by Marmer and his colleagues. Later, the bacteria were infected with sp8. As a result, the phage DNA was injected into the bacterial cells. Transcription within the infected bacterial cells produced radioactively labeled RNA complementary to the phage DNA. This newly synthesized RNA was isolated from the cells. Secondly, DNA from fresh culture of sp8 was isolated and the two strands were separated into heavy and light strand of DNA respectively. The radioactively labeled RNA obtained from the infected bacterial cells was combined with the heavy strand and the light strand. Hybridization was observed with the heavy strand. However, when radioactively labeled was combined with the light strand, no hybridization took place. Hence, evidence was provided by Marmer and his colleagues which proved that RNA is transcribed from only one of the DNA strands in sp8. Heavy strand acted as the template in this case. The newly synthesized RNA strand was complementary and antiparallel to this strand, having the same polarity and base sequences as that of the non-template strand, with the exception that thymine in DNA is replaced by uracil in RNA. Transcription unit. A transcription unit is a stretch of DNA of a particular gene sequence which is flanked by a promoter and terminator region upstream of the start site and downstream of the terminator region respectively. The three components namely are promoter, the DNA sequences, consensus sequences located in the 5' region which promotes, recruits or initiates the transcription process. These consensus sequences have been conserved throughout the evolutionary process and share homology in different genes of the same organism or in one or more related organism. It is present upstream of the RNA coding region or transcriptional start site. The promoter facilitates the binding of transcription apparatus to the DNA template and ensures that the initiation of each RNA occurs at the same point. Two promoter regions have been identified in the bacterial system: a Pribnow box or Tata box located -10 upstream from the site of initial transcription, TAAT sequence rich in adenine and thymine, and TTGACA located 35 nucleotides upstream. The specific sequence of the promoter is responsible for the binding strength of the RNA polymerase to the transcription unit. RNA coding sequence. The base pair sequence of the template DNA, which is copied in the RNA molecule. Terminator. The sequences of nucleotides which signals the termination of transcription and are part of the coding region. Types of eukaryotic RNA polymerase. RNA polymerase function location. RNA polymerase 1 function transcribes 28S, 18S and 5.8S rRNA molecules. Location nucleolus. RNA polymerase 2 function transcribes mRNA, snRNA. Location nucleoplasm of the nucleus. RNA polymerase 3 function transcribes tRNA, 5S rRNA and snRNA and few miRNAs molecule. Location nucleoplasm. RNA polymerase 4 some siRNA in plants. Location nucleus in plants. RNA polymerase. The enzyme RNA polymerase catalyzes the process of transcription. Comparison of structural composition of prokaryotic and eukaryotic RNA polymerase. Bacterial RNA polymerase. A single large multimeric RNA polymerase catalyzes the process of bacterial transcription. The RNA polymerase consists of a core enzyme made of 5 polypeptides, which are 2 alpha, 1 beta and 1 beta dash bound to another polypeptide called the sigma factor. The sigma factor recognizes the upstream -35 and -10 regions of the promoter and makes sure that this binding of the RNA polymerase and DNA is stable. Core enzyme can carry on the process on its own back and lacks the ability to bind at a specific promoter region. The binding efficiency of the RNA polymerase varies according to the actual sequence of the promoter region. The polymerase binds to the promoter region while the DNA is still in the double helical form, known as the closed promoter complex. Holoenzyme is the actual functional enzyme which helps in the opening up of the DNA helix by melting a short stretch of the DNA helix. This untwisted form of the promoter is known as open promoter complex. The sigma factor can leave the transcription bubble once the RNA chain reaches 8 to 9 base pairs. The released sigma can be used for initiation by another RNA polymerase. Further elongation of the RNA chain can proceed without requiring the sigma factor. The core enzyme can bind to the DNA molecule with the same affinity at any position. The binding sites for core enzyme are known as loose binding sites, but the holoenzyme binds to promoters very tightly with an association constant increased from that of core enzyme by on an average 1000 times and with a half-life of several hours. Eukaryotic RNA polymerase. RNA polymerase is large and complex enzyme. Example, yeast holoenzyme consists of 2 large subunits and 10 small subunits. Transcription elongation. Once the 10 to 12 long RNA is synthesized from the sigma factor leaves the transcription bubble, the RNA polymerase undergoes conformation change and is no longer able to bind to the consensus sequence. The core enzyme moving along the template, joining nucleotides to the RNA molecule. In the process, it untwists the DNA double helix downstream and then re-anneals it. The rate of RNA synthesis is lower than that of DNA synthesis, and an average of 30 to 50 nucleotides per second are added. Topoisomerases help in the uncoiling and recoiling of the DNA template as transcription proceeds. RNA polymerase has proofreading property which helps to remove any non-complementary base and continues the transcription. If the enzyme encounters a wrong base, it goes back and cleaves it to resume the synthesis in the forward direction. Transcription in prokaryotes. Initiation. The transcription apparatus recognizes and binds to the promoter region, which is identified by the sigma complex. RNA polymerase binds to the promoter sequence in duplex DNA or closed complex. The melting reaction to convert closed promoter complex into an open promoter complex. The polymerase pairs the base of the nucleotide triphosphate with the complementary base present on the template DNA. Termination of transcription. The sequences which code the termination of the transcription are referred to as the terminator sequences. Termination includes detaching of the enzyme from the DNA template and the release of the newly synthesized RNA molecule. Two types of terminators are present in the bacterial system, with or without an ancillary protein called Rho factor, namely Rho dependent, also known as type 2 terminators, and Rho independent terminators, also known as type 1 terminators. A polycistronic RNA is produced when a number of genes are transcribed in a single RNA. That is, a single termination site occurs at the end. Polycistronic RNA are absent in eukaryotes, and as each gene has its own initiation and termination site. Transcription in eukaryotes. Transcription in eukaryotes is similar to that of prokaryotes. However, it involves 3 RNA polymerase, which helps in recognition of specific promoter regions. These promoters or activators or enhancers have two regions, namely core promoter and promoter proximal region or regulatory promoter region, present upstream of a gene sequence. Sequence elements of a general eukaryotic promoter or gene. The core promoter is located upstream of the initiation site and consists of -35 to -52 base pairs. The Tata box, also known as Goldberg-Hognes box after its discoverer, is present -25 to -30 base pairs upstream of the start site, and consensus sequence is TA, TAAA. These promoters facilitate the formation of initiation complex and affect the rate of transcription. The regulatory promoter are located upstream of the core promoter. Example, cat box CAAT sequence 5-GGCCAATCT3- GC box sequence GGGCGG, centered at about -75 to -120. Any mutation which takes place in this region markedly decreases the rate of transcription, indicating their role in the efficiency of the initiation complex. Function of the general transcription factors. General transcription factor function in transcription. Transcription factor 2T composed of Tata binding proteins TBP and TBP associated factors. Function in transcription recognizes the Tata box in the promoter region or core promoter binding factor. Transcription factor 2B interacts with Tata binding proteins of transcription factor 2T and stabilizes Tata binding proteins. Transcription repeat Tata binding proteins Tata complex. Recruits binding of transcription factor 2F RNA polymerase complex. Transcription factor 2H function in transcription. Helicases activity for opening of the promoter complex initiates transcription. Enzymatic activities of DNA helix and ATP kinase and repairs DNA damage by nucleotide accession repair. Transcription factor 2A function in transcription, stabilizes TBP DNA binding, transcription factor 2F, binds to RNA polymerase and prevents it from binding to non-specific DNA binding sites. Transcription factor 2E function in transcription helps in maintenance of initiation complex and switching to elongation process. Transcription in eukaryotes initiation. General transcription factors GTFs have replaced the sigma factor of prokaryotes. Transcription factor 2D is the initial committed complex which recognizes and binds to the Tata box with the help of its TBP, that is Tata binding protein. The binding of transcription factor 2D facilitates the sequential binding of other general transcription factors, transcription factor 2D followed by transcription factor 2A, transcription factor 2B, transcription factor 2F accompanied with polymerase, and finally transcription factor 2E and transcription factor 2H and RNA polymerase to produce the initiation complex. Transcription in eukaryotes. Termination. Synthesis of poly A tail. RNA is released which destabilizes the RNA polymerase and DNA complex. Allosteric model. Due to destabilization, DNA and RNA polymerase separate from the growing RNA RAT1 exonuclease binds. Binding leads to a torpedo-like action which ferociously cleaves the RNA, leading to separation of DNA and RNA polymerase. RNA splicing. Self-splicing introns which are present in some rRNA genes. The self-excision involves an interaction between a guanosine cofactor and the primary transcript. The 3'OH group of guanosine is transferred to the nucleotide adjacent to the 5' end of the intron. The newly acquired 3'OH group of guanosine on the left hand of exon and the phosphate on the 3' end of the intron form a bond. The intron is spliced out and the two exon regions are ligated, leading to the mature RNA. Lariat formation in spliceosome. Small nuclear RNAs or snRNAs or SNRPs. So students, let us summarize what we have learned in this module. We have learned that transcription is the synthesis of RNA from DNA. The genetic information is passed on to generations through the central dogma of biology, that is, DNA to RNA via transcription and RNA to proteins via translation. Prokaryotes have a single RNA polymerase catalyzing the polymerization of ribonucleoside 5'-triphosphates NTPs, and the growing chain is always in the 5' to 3' direction. Core polymerase consists of 2 alpha, 1 beta and 1 beta dash subunit. The addition of nucleotides continues until the polymerase encounters a termination signal. Termination is of two types: row dependent, in which the protein factor row terminates the chain, or row independent, in which a GC-rich inverted repeat results in the formation of a segment of RNA that can form a stable stem loop structure by complementary base pairing. In eukaryotes, transcription involves 3 RNA polymerase which help in recognition of specific promoter regions located upstream of the initiation site and consists of -35 to -52 base pairs followed by a Tata box. The general transcription factors and RNA polymerase bind to produce an initiation complex. The RNA transcript has a length of 25 to 30 base pairs which keeps on elongating as new nucleotides are being added to the 3' end. Transcription via RNA polymerase 2 typically terminates about 500 to 2000 nucleotides downstream from the poly A signal either by allosteric model or torpedo model, which disrupts the transcriptional machinery and terminates transcription. The eukaryotic genes consisted of coding and non-coding regions which are joined by the process of splicing 3' to 5' modification of the mRNA to provide stability to it. The 5' cap is easily recognized by the ribosome which binds to it and initiates the translational process. Polyadenylation provides stability to the RNA molecule, that is, protects from exonucleases and a longer time period to be available for the translation process. Thank you.